Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this section to discuss NCERT Class 12, the chapter is Biotechnology Principles and Process. Part 5, Vector for Cloning Genes in Plants and Animals. You may have been surprised to know that we have learned the lesson the transgenic genes into as in plants and animals from the bacteria and virus which have known as in the age how to the deliver genes of the transfer eukaryotic cells and further to what do the bacteria or virus wants. So once you get observed to as an how to the genes will be get transferred when you say like as a bacteria and viruses, when you see how to the genes are get transferred into as an eukaryotic cells also. See they give an example. For example, here Agrobacterium tuberifensis. It is a pathogen of the several dicot plants able to deliver the piece of the DNA known as a tDNA. Transfer DNA to transfer as a normal plant into as a tumor and detect this in a tumor cells to produce as a chemical required by as a pathogen. So mostly the agrobacterium get using as a vector. That vector get help to as a get control disease in some pathogenic plants. Similarly, here when you observe to as an animals like similarly retrovirus in animals, they have as ability to transfer as a normal cells in cancerous cells. So retrovirus in this mostly virus here also same you get using as in some vectors. They also to get completely controlled as in some cancerous cells also. Okay, a better understanding of the art of delivering the genes, the pathogenes, their eukaryotic host has in a generated knowledge to transfer these tools of pathogen into useful vectors in delivering genes insert into as a human. So mostly this kind of these things we are using nowadays we get directly introduced into as a human cells automatically to get controlled as a the number of pathogens. Okay, this is now this tumor including the TI. Here TI means see here the now tumor that is including TI plasmid. TI plasmid. Here the Agrobacterium tuberifensis has now been the modified into as a cloning vector which has the more pathogenic to be helped to the plants but still able to use as in the mechanisms to deliver genes our insert their interest into as in a variety of plant. So nowadays we are using at this kind of number of their pathogenic plants. Still some genes are introduced into as in a plants to get controlled to as in the number of pathogens. Okay, similarly retrovirus also have been their dissimilar and now used as in the delivery of the desirable genes into as animal cells. So like how do we are introducing it as in plant cells, here animal cells also we are introducing as in some retroviruses. And sorry. So once a gene or DNA fragment has been ligated into as a suitable vector, the transfer into as a bacterial cell or plant host where the multiples. So once vector get using what happened, they produce as a multiple copies within a short duration period. Okay, these are completely vectors. Let us discuss in a competent host from transformation with a recombinant DNA. Recombinant DNA. Let's see. Competent host for transformation with recombinant DNA. Since the DNA is very hydrophilic molecule, cannot be passed through cell membrane. So it is a hydrophilic, it is not a pass through as in a cell membrane. Why? Because in order to force the bacteria takes place plasmid, the bacterial cell must the first made up of as a competent to take up DNA. So it is a competent host cell. It is does not uptake to as in a what the total host organism into the DNA. This is done by as in treating with them with help of in specific concentration of in a divalent cation such as in a calcium, which there increase the efficiency with the DNA enters the bacterium through the pores of the cell wall. So easy to get bacteria to get enter into as in a bacterial pores or in its cell wall. The recombinant DNA can be that forced into such as cells that incubating. First one is what? Incubation process. Incubating within the recombinant DNA on ice. So first the DNA we get keep on the ice incubating, followed placing on the briefly 42 degrees heat shock, that is called as a heat shock treatment, and then they're putting off back on ice, and then after their heat shock treatment the DNA is again to get keep on the in a ice. This enable to the bacteria to take up the in a recombinant DNA, then only the bacteria will be the uptake to as in a DNA. This is not is only one way introduced LNDNA into as a fossil. It is not a single step. It is in a the repeated process. In this method known as in a micro injection method. It is what? Micro injection method. The recombinant DNA that directly injected into as a nucleus of any animal cells. So we are directly introducing into DNA into the animal cells. Okay, animal cells. Next, the another method it is in a suitable for as in that cells are recombinant in a bombard with as in a high velocity micro particles of gold tungsten coated with as in a DNA method known as in a biolistic or gene gun method. So another here mostly when we are observed to as in some plant in a bomb they completely blast in that bombard method in high velocity we using as in a their golden tungsten their particles we are directly to DNA introduced into as in a what plant cells. This process called as in a biolistic method or in a Jingan method. And this last method of using in a dismayed pathogen vectors, the dismayed pathogen vectors, that is what which is when allowed into the infect the cell, the transfer in a recombinant DNA into as no cell. Directly we are introduced into as in a host cell. This is in a last method. Now that we have been learned about as I told for as a contrasting recombinant DNA. So this process we are learn tools of recombinant DNA process. Let us discuss as a process of they are facilitating recombinant DNA technology. Let's we discuss about as what in a completely process of in a recombinant DNA technology process. Okay, remaining we discuss in next class. Thank you for watching. Please share, like and subscribe. Thank you.